Good afternoon. Okay, so I'm going to start by talking about recent effort in engineering a tRNA near-chromosome yeast. So my group is part of the Wellcome Trust Center for cell biology in Edinburgh up to now. So my group has been working closely with BGI in the last few years. So we recently published chromosome 2. So we just finished the synthesis of chromosome 2. We just recently completed another chromosome, that's chromosome 7. But I'm going to talk about the smallest chromosome in yeast today. And this, by the way, this is a consortium I started when I was a postdoc with Jay Booker in Johns Hopkins a few years ago. And this is a great consortium I have, the privilege to work with in the last 3-4 years. So the tRNA near chromosome, as you know, we remove all the tRNA gene from the genome. So we remove all the 275 tRNA genes. But tRNA is essential, right, because they need it for translation. So what we have done is we remove all the tRNA genes, relocate them into a new chromosome we call party chromosome, so they can do whatever they want on the chromosome. So this is, the shortest chromosome in the yeast, SC2. So it's only 180 kb, but as a hybrid of multiple yeast species, so only 10% of the sequence directly derived from Saccharomyces cerevisiae. And so I argue this is the most orthogonal chromosome in the yeast. So this work was largely completed by Roy Walker, who synthesized the chromosome. Now a new postdoc in my group, Daniel Schindler, is going to characterize the performance of the near chromosome. So I'm going to talk about design basis, right? So, instead of having a natural template, you can start doing your design. We start from scratch, right? So there's no natural counterpart to design this near chromosome because, this kind of chromosome never exists in nature, as far as I know. So this is a chromosome without any pol2 gene, only pol3. So the first thing we did is we look for centromere, right? So centromere is needed for kinetical assembly, and this is where microtubule is going to attach and during the cell division. For yeast, it's easy because we have point centromeres, so it's only roughly 125 base pair, and we took it from chromosome 6. But the problem is for higher eukaryotes, many of them require regional centromeres, and that's not sequence defined. It's more epigenetic defined, right? So sometimes you're talking about 100 kb for centromere, and it's not well defined. But we're lucky we got this point centromere to start. And the second thing is we need replication, right? So we need to replicate the chromosome. So we took our sequence. So our sequence actually also quite easy in yeast. It's only 11 base pair to 17 base pair. It's very T-reached. And this is required for the OC complex to bind throughout the cell cycle. And for many high eukaryotes, it has many origins, but not all of them are activated. So we put 4 origins, and these are origins we took from Candida, right? So we took origins from Candida, and they can fire early and strong in cerevisiae. So we put them, roughly 40 kb each, from each other. The third thing we need is telomere, because telomere will protect the chromosome from shortening and chromosome fusion. And there are roughly 300 base pair, and which is TG rich. So we took this from Leslie Mitchell's telomerate. So basically, she designed a construct which she embed 2 telomere C sequence embedded in actin-1 intron in the ULA3 gene. So you can embed this telomerate in different locations in the circle, so you can permutate. You can open it up in different permutations. And telomere is important because in high eukaryote, it's shortened because incomplete replication of the chromosome, and also Okazaki fragment need to attach the RNA primer need to attach the locking strand. So we got that. So we're going to open up this circle at different permutations later after we complete. Then we also look at TER size. So because the chromosome is going to replicate quite like crazy because it's just tRNA genes in a chromosome, right? And so TER sites exist in RDN locus to protect against replication stress. So these are the sites, fork1 protein is going to bind and store the replication fork, right? In observed cases, and that ensure the replication fork to go in the same direction as the transcription. So you can imagine the transcription and the replication fork is going to go towards each other and get stored at the TER site. Right, so in a nutshell, what we have done is we took 275 tRNA genes, and tRNA genes are really small, they're 75 to 91 base pair, but we flank it with 500 base pair flanking region, and 40 base pair 3' flanking region from the other yeast species called Ashibia goldsby. So this is really remarkable yeast species, has no transposon at all. So to answer earlier question, what's the basis of transposon is not essential. There's a eukaryote without any transposon. But they have really high gene centenary to cell VCA. And so we designed roughly 600 base pair. And then we flank this tRNA cassette with a pair of ROX sites. So these are conditional recombination sites, which are modulated by a recombinase called G. And then we design them chromosome by chromosome. We call them tRNA arrays, so we design them in the same direction, so all the tRNA cassettes pointing to the same direction. And different chromosomes have different numbers of tRNA genes. And we pair them up to roughly 20,000 base pair. So for instance, we pair up chromosome 1, chromosome 2, and chromosome 3. So they make up roughly 20,000 base pair. And then, for instance, the chromosome 7 we have has the most tRNA genes, so SF make up almost 20,000 base pair. So they point to each other, so replication fork run into each other and then arrested by the TER site. And then we put in this R sequence I mentioned, which we get from Candida, so we can evenly replicate the chromosome. And so this is the design for the near chromosome in a nutshell. We synthesize it. And then this is evidence the design is going to work. So there are 3 single copy tRNAs. We knock them out, and then in all cases, our tRNA cassette were able to complement the deletion. But to profile the tRNA expression in a whole genome manner is difficult because tRNAs are very rich in secondary structure, and they're also very rich in modifications. Until very recent, there's a thermostable group 2 intron reverse transcriptase being identified, and people develop methods to do tRNA-seq. So normally when you do RNA-seq, you throw away the tRNA genes from get-go. But we have been working with Lars Neimer's group in EMBL to do tRNA-seq, so we can high throughput profile the tRNA expression. And we also work with David Torvey's group in Edinburgh to do the CRUC, which is to map the RNA and protein interactions. And this is a rather busy slide. I don't have much time to explain, but the key conclusion is tRNA near chromosome expressed really well. So every single tRNA expressed just like wild type. And that tells you these are really robust genes that are very context independent. And the post-3 occupied the near chromosome, just like wild type. There's no problem. And I did not tell you in the designs, we take out all the introns, right? So yeast has not many introns, but tRNA are really rich in intron. So we take out all the introns, right? So after you take out all the introns, all the tRNA expressed just like wild type. So splicing is not essential. And the other thing we found quite interesting to mention is when we finish chromosome 2, right? And then we have a version of chromosome 2 sent to without any tRNA supplement, right? So we delete all the tRNA. And we have another version of the synthetic chromosome 2, which we supplement tRNA array. So what we have seen is when you delete all the tRNAs, we see a upregulation of amino acid synthesis, and we see a downregulation of translation. But as soon as you put in the tRNA array, that revert the phenotype. But we are not sure well this is a global manner, so we are trying to do it with every single chromosome we have, and to see whether this is something would be interesting to look into further. And the other thing we are quite interesting is the moonlighting jobs of the SEN complex. So I told you, we remove all the introns, right? So maybe I take a minute to remind you about the life cycle of the tRNA. So tRNA are transcribed in the nucleolus and then got processed in the 5' and 3'. But it needs to be exported to the cytoplasm because there's a complex called SEN complex as on the surface of mitochondria. And only yeast is on the surface of mitochondria. And what it does is splice off the intron in the tRNA, and the tRNA got charged and re-imposed back to the nucleus. But, for other eukaryotes, the SEN complex is actually not in the cytoplasm. Let's suggest this SEN complex in yeast is doing some moonlighting job. So now we have no intron. Splicing is not essential. Now we can go in and knock out the SEN complex and try to identify what other moonlighting job is doing. And my time is running out, but I would like to tell you the other thing we're looking into at the moment with this near chromosome is, we're looking into the localization of tRNA. So there's a long-standing question about where tRNA goes. So one paper says it goes to nucleolus, the other one says it goes to nuclear pore complex. Now we put them together in the chromosome, we should be able to follow their localization. So I guess that's that. So with that, I would like to thank my group in Edinburgh. So this is my group in Edinburgh. Thank you very much. Yep.
Was the SEN complex still essential?
We don't know yet. Yeah, we tried to knock it out, but it doesn't seem to be trivial to knock it out.